Hello, good evening, everyone. So today in this week 6, we will be looking into two most techniques of what we are looking at for studying the interactions between of different biomolecules. The first thing is one is which we will be looking into it. One is the high throughput arrays, among which one is label-based techniques, but different kind of array of which we had talked about and which we have seen obviously in the previous uh, classes as well was of the reverse phase protein arrays and one level free technique which is SPR or the surface plasmon resonance. So now reverse phase protein arrays of which we have started looking into it from the previous week in a much more detail, among which one major thing during the different protein arrays, one is that the way it can have for the high throughputness of this particular technique, which led us to uh, get assay of the wide variety and a wide range of proteins for that matter in a biospecimen to monitor their role and their particular how much they have been expressed over different conditions, but among which reverse phase protein arrays. So we had also looked into NAPA, which is the cell uh, which does not include the tedious process of expressing proteins and then purifying. And in the reverse phase protein arrays also, the major thing that has been changed is that the cell lysates are immobilized and does not require immobilizing any antibody or any protein for that matter on the arrays. So this is also known as the micro western blotting on the basis of the technique, because it uses a similar kind of principle of whatever is used in the western blot to uh, for uh, validating any protein using antibody based method from a cell lysate or tissue specimen. And here the major things which we have also looked into the previous week is that the having the printed cellular lysates over this kind of channels, and it's captured with the antibody and the secondary antibody. So the two major things which we can talk about uh, for reverse phase protein arrays, or which is known as RPPA in short terms. So this is one is the label based. So it is a label based technique because it is captured with the antibody and secondary body which are conjugated with the label. And second thing is that the cellular lysates or the tissue lysates, in the same way, if you want to do Western blot or serum, and you can print and take it. So this is known as the particular micro Western blotting. So this is a major difference again between the forward phase protein arrays and the reverse phase. So here you can see that particular antibodies are printed. So you need an antibodies, you need the protein. Say you will pass on the cellular analytes, and then finally the secondary antibody, which is conjugated with particular tag, and which will be recorded for you to understand that particular uh, for that particular antibody, that particular protein is how expressed or how much it is expressed, and uh, all those things. But the problem with this kind of forward phase protein microarrays that you need to have two antibodies to different two different sets of antibodies which you need to take it, and then finally go for your particular analysis. So this is the major problem, not problem, but one limitation or drawback it has. But in the reverse phase protein arrays, you need to have only one antibody and don't need to immobilize any further protein in this membrane, just the cellular lysates or the tissue lysates, and which will be captured by the antibodies. So for which particular antibodies you are getting that particular signal, then we can monitor that. So now this only draws us to the fact. So if we now think of doing a very robust experiment, so if we just have a very robust experiment stating for RTPA, so what are the two things that you will require? Two things. If I ask you that, what are the two things you need to be very careful with? You need to be very careful. One thing is that definitely the samples. The samples should be very neat and clean because you are printing the samples on this particular chip. And second is the antibodies which are getting detected. So these are the two important things which you should be very much careful of. Otherwise, you won't be able to perform this particular kind of experiment in the experimental side point of view. And now, after the data is generated, you need to have a robust data analysis pipeline. Otherwise, whatever you get it, so you won't be able to get this particular data set or particular thing if we don't have the robust data analysis pipeline in place, which would be leading to the difficulties. Otherwise, you will be facing the problem. So one thing is that the sample preparation and cleaning up of the samples for you to print on these particular chips. Then is the antibodies of good quality and high quality, of which can able to detect specific proteins, not no non-specific proteins or the non-specific signals. Third is that after the signals have been captured, we should have a robust data analysis pipeline. So this only will give us the opportunity to have a set up a very high throughput, reproducible and robust reverse phase protein microarrays for getting information starting from protein-protein interactions, protein post-translation modifications to understand in different samples. So these particular things should be there in particular thing. Otherwise, we won't be able to deduce all that experiment and deduce that thing. So this is quite important to understand, and which we will be looking in subsequent slides, that how these particular things are to be ensured for the sample, what kind of samples we can take, what are the lysis buffer that needs to be taken care of, that which are compatible and don't interfere in the signals and the backgrounds, and also the efficiency of those extracting the proteins should be pretty high. Third thing would be the antibody selection, and then finally the data analysis. So all these things we would be seeing in the subsequent slides to get a better understanding and to better understand. So what kind of samples that can be used for the reverse phase protein arrays? So as I mentioned that all samples like starting from tissue, so you can have a chunk of tumor tissue along with some matched control tissue. So matched control tissue or a tumor tissue, you can take that particular thing. you can prepare lysate. So you are just extracting the proteins out. Similarly, cells can be any cell lines which you are maintaining can be of a tumor, cell line. It can be a primary cell line. It can be very few cells like stem cells. So if you have taken the stem cells or certain kinds of cells which are very few in number, those can be taken forward, and with this particular lysate preparation and all, you can perform the analysis. Then is the biofluids like serum and plasma. So you have an excellent opportunity to work with this serum and plasma, which uh, not so invasive but still quite easy to take out. And then finally you have to also are indicators of various markers and various markers for various diseases. Then biofluids like saliva, saliva, CSF, etc. The best is to have any kind of non-invasive. So if you have this kind of non particular things like the non-invasive ones, non-invasive specimens, so that would be a very good for the clinics and for the patients to have certain markers through these certain arrays which can be taken forward, or serum, plasma, tumor lysates, cell lysates, tissue lysates, and all these things can be used for reverse phase proteinase. So as I mentioned, the one of the major advantage of this reverse phase proteinase is that a wide array of samples or biosamples can be wide array. So wide array of biospecimens or biofluids can be used for these particular applications. Okay, but then what are the things that needs to be keep in mind? We will look into that. So just to summarize the reverse phase arrays, so this is high throughput technology. You can understand it's quite robust, can perform these proteinases on thousands of samples simultaneously. So it can occur on thousands of the samples simultaneously. So among the tissue lysates, what are the tissue lysates that you can use for setting up the experiment of reverse phase protein microarrays? So one is macro dissected tumor samples, laser capture micro dissected, and FFP fixed tissue lysates. So all these three categories you can take it forward. Sorry. Cell lysates, you need tumor cell lysates, small number of cells like stem cells, as I have already mentioned. Serum, plasma, body fluids like urine, saliva, or non-invasive. Okay, so all these things, samples can be taken in RPPA. So now, what, as I mentioned, that though wide array of samples can be used, so though wide array of samples can be used, need to be so need to be critical with the sample preparation. So the major first component is the lysis buffer. So why lysis buffer is very crucial? Yeah, so why lysis buffer is crucial? Lysis buffer is crucial because it lyses the cell membranes. And extracts out the proteins from cells. So that's why lysis is quite important thing here. Yeah, so it extracts out the protein. So it should be compatible. So it should be optimal for maximizing the RPPA signal. So it should be compatible with your detection methods. So be it colorimetry, chemiluminescence, or fluorescence, whatever methods we are using. So lysis buffer should be very much compatible with that. It should not interfere with the substrate on slides or absorption of those lysates with the. So the lysis buffer that we are taking, firstly it should give maximal efficiency in lysing and making the protein out, maximal efficiency. Now second is that it should not interfere with the substrate on slides or absorption of the particular lysates. And the extraction efficiency vary with different lysis reagents. Second thing is that protease inhibitors is a must, otherwise proteins will be denatured or degraded. So proteins should be protected. And then some common buffers that are used like PCCL, glycerol, STase, urea, chaps, HIPS, Triton X100. And all sample types cannot be solubilized by a single buffer. So this is the very important. So this is a very important thing to be considered, because otherwise what will happen is that we would try to solubilize all the samples using the single buffer and we don't work very well or effectively. So that's why we need to have different reagents and different buffers so that we can take ahead and we can measure and we can try to optimize for our sample type and our sample preparation, which we will be taking it forward. And then what we will do is that we will try to see that it is compatible with our detection methods and it is not interfering with the signals and whatever way we are detecting on colorimetric or chemiluminescence. Majorly, it is the fluorescence that is detected, so uh, it should not interfere with that. Now, the most so one important thing which I had mentioned here, one was the samples. That is most important because you have this particular membranes slides where you are doing the cell lysis of those samples, you are printing it. So samples is one thing that is quite important. Second thing is the antibodies. So samples, we have seen that the samples can be a wide variety of samples, can be a wide range of biospecimens, but the major importance should be given with the sample preparation. And for that, different lysis methods are there, different lysis conditions are there that needs to be optimized according to the sample preparation, samples condition, and the experimental demands. Second is the most important thing is in the reverse phase proteinase is for the antibodies to get robust protein, pick those robust protein, and to take it forward for further analysis and further taking of those. Now, what we are telling is that antibody validation using the Western blot. So, all those antibodies are validated using Western blot. So, how these antibodies are validation using Western blot means what is the meaning of this? That so which sorry so this if we tell antibody validation using Western blot, so which antibodies to choose or employ. Which antibodies which of the following antibodies to choose or employ for in RPPA experiment. So this is what the major question is. So that question is answered using Western blot. Using not Western blot, but there are a series of steps which we will be looking into it, how particular antibodies are chosen to be used for RPPA. Otherwise, it is not used. So this is the antibody. If I just into it, so if it gives Western blot with single sharp band, this is the first checkpoint. So this is the first checkpoint to go ahead of Western blot with the single sharp band. So if and only when single sharp band is visible for that particular protein of the said antibody, then only that is given a go ahead to for the second round of validation. Second is that Western blot happening with the pooled license of interest. So if you have the control the tumor, for example, so you are taking 6 sample and you are trying to do that. So there also single sharp band should come. So this also gives that the antibody is quite effective enough of capturing the particular expression, particular protein in those kind of lysates. First checkpoint, then is the second checkpoint. Then is to then is the third checkpoint. What third checkpoint is that? With some when we perform, if you perform some knockdown or some other experiments, there you are modifying the proteins, certain proteins. So modified your certain proteins. So when we are modifying certain proteins, for example, doing certain phosphorylation and all those things, then what is happening and how you are getting the modified proteins, and still your antibody is able to capture that or not, or to get that information. And then fourth checkpoint is that, so if all these checkpoints are crossed and we get a yes and the go ahead that it is fine, that the antibody is working fine each and every step, then only can be taken forward for further in real experiments. So all these checkpoints needs to be covered. So then is that validation by RPPA, which requires serial dilution of the pooled lysis of interest and can be taken forward. In the RPPS slide, when you have the serial dilution of why this serial dilution, so that lowest concentration proteins at limit so the limit to detect those protein concentration. So this is what makes it quite important for us to get it with so that lower to lower level of those things can also be taken forward and can be used for our particular analysis. So this serial dilution for this particular interest or for particular thing that can be used forward is used. So this is how uh, we can get this particular checkpoint for further analysis, so that these antibodies, once the antibodies is passing all these checkpoints and is given a go ahead, that all right from the starting point of the single uh, what I can say is that the yes, that from the starting from the western blot, that yes, it's a single sharp band, second lysate, third, fourth, and when it is getting validated finally by RPPA through the serial dilution of the pooled lysates of the interest, so then this particular will be given a go ahead to use in. Okay, so these are the steps for validation of the RPPS antibody. So one, we have looked for the sample preparation, which implies on the relies on the kind of the sample lysis buffer, which with the what we want is the maximal efficiency in extracting out the proteins and minimal interference with the signal. So if I just say for the samples, so it is that the maximal extraction of the proteins, minimal interference of the signal, and the western blot, and the antibodies is something is that minimal extraction of the signal with the samples of the lysis buffer that is being used. Whereas in the antibodies, to have a robust antibodies, if we try to say or search, it happens with that it is having a single rise sharp, then with the full lysate, then with the knockdown experiments, and then finally by the serial dilution of lysates to have that at what minimal amount concentration also is getting detected, and once every step is uh, it cleared in different the checkpoints, then only go ahead is given to use that antibody in RTPA experiment. So you need to invest a lot of time in validating those antibodies before you can perform the actual experiment. So that is one of the major bottleneck in this particular experiment, right? So now moving on, which is the preparation of our protein microarray. So now we are trying to prepare this kind of arrays of or the preparation of the protein microarrays, which we know to some extent. One is obviously the contact microarray method. Sorry. So it's the contact microarray method. So if you have those microarray which you're contacting or physical contact is there. So that is the robotic method or the using the robotic arms. Now this is for the contact microarray method. Others are the non-contact microarray methods where it doesn't lead you to contact with the microarray methods. So one is the ink jetting method, electric spotting, photolithography. So these are the non-contact microarray methods. So if we say protein microarrays, one can be printing, can be contact, very popular, and then non-contact, which is emerging. In the non-contact method, what are the steps or these things are there? Injecting piezoelectric or photolithography. Now, what are the commonly used array surfaces? So that is also play a role in the absorption of the lysates. So this surface will lead you to have the absorption. So these are what needs to be kept in mind when we are doing. So these are the some of those different commonly used array surfaces, which we again need to be careful in choosing because of the experimental demand and the sample type of what is getting absorbed and which we have certain reactivity and the surface absorption, etc. We need to decide among which nitrocellulose is one which is being mostly popularly used, as well as gold is also being used due to its low reactivity nature, because it will also depend on the backgrounds. So this is also quite important to get to know. So among the which common which you are very much familiarized with it. First thing is glass, second gold, nitrocellulose, and hydrogen. So these are the uh, surfaces. So these are the surfaces to keep. In the lysats. So having moderate reactivity, gold is having low reactivity, nitrocellulose moderate reactivity, hydrogen moderate reactivity. Glass has low surface absorption, but nitrocellulose and hydrogen have high surface absorption, but mostly the gold or glass are also used, or nitrocellulose is also used very much for this kind of application, specifically in the field of reverse protein arrays. So, what is the antibody signals detection means for the reverse phase protein arrays? So, I am repeating once again, for reverse phase protein arrays, two things which is most important is the samples. So, the samples should be prepared in such a way that it has the maximal efficiency of extracting out the proteins but minimal interference with the signals. So for that, different kinds of lysis buffers are used. Some of them are common, some of them are used specifically for particular sample type because all sample types want to behave the same. Second thing is antibody. Antibody is again validated using four different checkpoints which it goes through it. One is for the identification through Western blot showing proper band. Second thing is that for the pooled lysis. Third is for the your with the knockdown and the modified proteins with certain modifications of, because our aim is to identify those proteins as well with modifications, and to get that what which also we can use, like if you try to mimic and try to find out the phosphorylation pattern between your diseased and the normal state, so you can use phosphatase inhibitors as well apart from the uh, protease inhibitors, so that also can be used. So this is quite important. and then finally validation by RPPA serial dilution that have been pulled lysates for together, which you can take it forward, and then only you can give go ahead for that particular antibody to use against against any kind of detergent or anywhere where they want wish to take it forward for use. So these are the important things. Next important thing what we was about to mention is that not only these two methods are important, also this the chip where it is there also used with different surfaces with different properties like moderate reactivity or low reactivity or high reactivity or low surface absorption or high surface absorption. So among which like the nitrocellulose and the glass slides are mostly used. Okay, so which are mostly used for further analysis and further taking forward. So now what does this antibody signals detection means for RPA? So these include the sensitivity and the specificity of the primary antibodies. So how strongly it can detect the signal from weak signals. Okay, so signal detection with to be crucial with no protein separation possible. So the major problem is that so just think of whenever we have, for example, this kind of microarray. So you have this proteins. So if you are using cell-free or cell-based system, you have those proteins which have been separated out. Now for RPPA, what you are doing? Here's the cell lysis. So there's no separation of the proteins. So in this RPPA, what is happening? Cell lysis are printed directly on the chip. So there is no protein separation. So it is not giving any kind of protein separation for us to take it forward. So that's why it becomes very important and crucial to have a signal detection, robust signal detection method in place, because we are detecting from the cell lysis which doesn't have any particular level of expression for, and it has basis of the local of those particular cells, and on the different expression levels it will be expressing around, and also in the disease condition it might be differently analyzed and differently done. So it becomes quite important to have those particular thing. But in other techniques, what you are doing is that you are separating the proteins out. But here it is not so the case. That's why it becomes quite important to have very good signal detection methods. Now, what further can be used as, like, the proteins can be expressed of the low levels, and the visualization that is there, that will definitely require certain kinds of amplification. Okay, so these proteins are expressing of the low levels, and this will require the amplification, because the proteins, as I mentioned, that if you are doing with the different lysates which are printed, which you are not separating out the proteins, and different samples in a high throughput manner when you are trying to do, so the proteins that can be are generated can be expressed in the low levels. So that's why you need the amplification, and the signal detection methods are to be crucial, because it contains no protein separation, and obviously the specific sensitivity and specificity of primary antibodies of there are different checkpoints through which it should go for giving a go ahead to using actual RPPA experiments. So tyramide signal amplification, or the TSA, is used to increase the sensitivity of these reverse phase arrays. So this is known as the TSA. So it is kind of an epitope that gets attached to the place where the interaction happens and tries to amplify the signal. So this is known as a dynamic signal amplification. So polarimetric or chemiluminescence has problem in the linearity of the quantification of the signals. So fluorescence based signal amplification methods are generally used to avoid issues with the signal detection. So generally, as I mentioned only in the previous thing, that mostly that it is being used is the fluorescence based signal amplification to avoid issues with any kind of signal uh, detection. So again, if I just summarize you all for this slide, what is the major important thing is to have, and which I think you have understood from the experimental setup. If we are doing this kind of particular bringing out this kind of particular experimental setup, then definitely we should uh, consider giving a thought and giving a shot to have particular like to have a robust antibody. So once we have gone through the sample and the antibodies that we have given a go ahead with and have passed or crossed all the checkpoints, then definitely it should have crossed as well as to include the sensitivity and the specificity of the particular primary antibodies, and then the signal detection methods needs to be crucial because it does not allow any kind of protein separation, which is easily done on the different other platforms. So that's why proteins may be expressed at low levels, and that's why visualization might be requiring certain kinds of amplification. Then dynamite signal amplification is used to increase the sensitivity of the reverse phase protein arrays, and colorimetric or chemiluminescence is something which is have certain kinds of limitations in linearity for the quantification of the signals. That's why fluorescence based signal amplification methods are used, which avoid issues with the signal detection. Right now, next part. So again, I'll just give you in the reverse phase protein arrays. One thing is that so three things needs to be kept in mind. One is samples, wide variety, but preparation needs to be done with great care because it might have include certain lysis buffer which might interfere and the extraction efficiency of the proteins. Then use of protease inhibitors and the phosphatase inhibitors are a must if you try to uh, identify certain proteins, if you try to identify some phosphorylations and all those things. So samples are very, very important to have the wide and but the major advantage is that any kind of biospecimen right from tissue lysate, cell lysate with minimal number of cells or very less number of cells can be taken forward and can be used. Second, that is used for your thing is the antibody, which will go through different cross checks, for cross checks, which needs to be gone through four different cross checks before going to the final uh, analysis. That is, before you go take the further analysis, and if you giving the go ahead, then but also the signal detection system must be robust, and you should amplify certain things for the low sample, and there's no separation of the proteins on, and is direct the cell lysate or the tissue lysate. So that's why you need to have gone through this. And third thing what we had talked about was the data analysis. Now of what we would be discussing about is the from the data analysis. So once you have got the amplified and all those particular values and particular this thing, then what it needs to be done, how the data analysis needs to be done, how the normalization is to be done, and to get the actual values and compare it in different samples. So you have the raw data with the quality control, and then you are doing the data normalization, then different plots to find out the difference in expression in different samples for particular protein. For particular protein, then you have the signaling dynamics, and then the cluster analysis and the heat maps, of which we need to understand that how particular samples are clubbed around, and some features or the certain proteins which are showing the different rates and different those things. So now different kinds of normalization that is there is this kind of normalization can be used, but all this normalization will give you different kinds of at different kinds of dimensions to your data. So you need to be very crucial in understanding that which kind of normalization will suit your data best. Either supercurve normalization, total protein normalization, loading control, variable slope normalization, robust Z normalization, housekeeping Z normalization. So you are taking that particular gene's value will remain the same across the samples because it's a housekeeping gene, and whatever variations you are getting, you are trying to, on the basis of that, you are trying to lower or increase the intensity of other proteins as well, which will lead you to the on a same scale to uh, compare between the two different samples, then invariant protein set normalization or the global median centering. So this is the major different kinds of normalization techniques that you need to use of keeping in mind of whatever you are trying to do. So why normalization is required, or why is this? It is very much essential because you are here. We are just the right from the beginning huge variability. So what are the variabilities that are associated in our PPA? Variabilities. So what are the variabilities that are being associated with RPPA? So first is that cell or the tissue. So if you just think of using cell as a sample which you are comparing between uh, control cells or two different cells, or you have done certain kinds of treatment of the cells, or something like that, or stem cells from normal and the diseased person, etc., etc. So the thing is that variabilities that are associated right from the extraction So you are extracting the proteins out of it, which needs to be extracted out, so that the amount of protein extraction is different. Then you don't have any control over that, the amount of protein extracted and the lysis buffers, then amount of protein that have been during the preparation of the array on the membrane, and the uh, absorption. So all these things are very much not in our control, and is which are the uh, places where variabilities gets introduced in RPPA. So it is essential to account for the differences in the total protein concentration. Each tissue array on the sample needs to be compared directly. So normalization is based on total protein per microarray spot. Amount of protein present per microarray spot is what we count for the normalization, and the normalized intensity values measured by dividing the measured intensity value of the antibody by the corresponding measured intensity of the total protein, that how much intensity values are there uh, divided by the measured intensity values of the antibody in the corresponding of the total protein. So this is how you can getting the uh, normalized values, that how much of the antibody and how much of the total protein. Now there is an important concept is to have negative control slides. So what is negative control? Negative control is that for the non-specific binding of the it is for the non-specific binding of the secondary antibody to the array, which is for the so when just think of the experiment, you are having the cell lysates, you are giving primary antibodies, right? So it is going and detecting with that particular protein. Now you have secondary antibodies which are conjugated with some fluorescence level, and it is getting detected. Now in this full array there can be secondary antibodies getting non-specific bind, non-specific non-specific binding, right? So you are getting non-specific binding of those particular secondary antibodies. So we have to keep one slide where we just without the primary antibody, without the primary antibody, what we are doing is that we are utilizing the giving the secondary antibody. So now wherever it will get attached to it, So wherever it will get attached to it, we will get to know that whatever secondary antibodies are doing. So in this way, what we are getting is that we are having a different uh, nature or different things, different what we can what I want to say is that we are getting that false positive result basis of the non-significant binding of the secondary antibody. So this is the concept of the negative control. But wherever we will get these particular signals, we will subtract that value from the our original experimental slides so that we can normalize that, and which will lead us not to have any inconsistent data, as well as not lead us to some incorrect data or inaccurate data. Because if we are claiming to have certain proteins which are important and which needs to be looked upon for, and which needs to be looked upon for uh, particular things, then definitely it should not be taken in a way that we get some incorrect data due to the non-specific binding of the secondary antibodies. That's why it is a negative control slides that is required. Now, so if we talk about the applicability of the reverse phase proteinase, so in the various types of samples, so as we have already mentioned, that can be any kind of biospecimen, so any kind of biospecimen, which can be tissue, which can be cell lysate, which can be saliva, which can be serum plasma, high sample throughput, low amount of sample required, low amount of sample, but antibody signal detection and those things should be very much robust, and some kind of amplification for visualization of low abundance proteins, and it should be high sensitive in nature, and the nodes of the signaling. This is because the PTAMs and the signaling networks you can understand through easily. Okay, so this is for the RPPA or the reverse phase protein arrays. Okay, hopefully you don't have any confusion. So here major important thing is for the samples, that kind of lysis buffer that is to be used, because you know that if we don't because it should not interfere with the signals with a huge amount of uh, good amount of protein extraction, then we should have a certain then the antibody validation, then the data analysis, and also the major thing is that also one thing I forget to mention, that absolute quantification is possible through RPPA. Absolute quantification possible through reverse phase protein arrays as well. And the data analysis, of which RPP analyzer is one of the most robust data analysis technique that have been developed for analyzing this kind of data. Second is the data normalization is to be done because of the variability of huge amount in the data set on during the generation of the data. So that's why those things are pretty much required, because that will give you an idea of how to take it forward and how to perform those kind of analysis. Then you have uh, you can do the absolute quantification by gradually increasing the concentration of the proteins and measuring the signal intensities, and then measuring the unknown signal, and on the basis of the line of curve you can do that. Then different PTM signaling you can do phosphorylation, but also to keep in mind to use PTMs and kinase inhibitors, uh, PIC inhibitors, phosphatase inhibitors, etc. And high throughput technique. It is definitely a high throughput technique, and also it requires a low amount of sample. So now moving ahead is that if which we have also talked about to some extent in the first lecture about these two hybrids and the uh, PET system, and then affinity chromatography, immunoprecipitation, etc., etc. Some of the traditional approaches that are used for studying the interactions, and the high throughput approaches are always like the protein microarrays and the level free technologies. So, what are the different detection platforms that are used? So, again, so now we are moving on from the one kind of technique of the reverse based protein arrays, which uses a level based technology, and though it is a high throughput technique and is used in a much robust way, but the different and also the protein microarrays and other approaches which uses the level based techniques, so there's some problems in the sense that with the labeling of the query molecule with the marker track, that is for the fluorescent, as for example that the KB luminescent and the colorimetric methods uh, poses a problem, as well as with the labels. Then there might be some insignificant binding, non-significant binding, and giving certain results. So there are some difficulties. So anyway, there are this is one of the most popular methods. So apart from level based, so there is another method which is for the level free, which measures the inherent property of query, which is includes your masses, dielectric property, etc., etc. And level free methods are the ones that allows you the real time measurements and avoid the issues related to the tags or fluorescence, whatever is done. So that issues are neglected, that issues are not taken care of, allows for the real time measurements, and it also measures the inherent property of query, example the mass, dielectric property, etc. So one is the level based and another is the label free detection platforms. So what is the now after, so we had detailed in we have discussed in detail the label based techniques, among which was the cell free methods like NAPPA, among the protein microarrays and the reverse phase protein arrays. So now we are moving on to the label free measurements, which will give us an idea about this what how this interaction is happening, how what is the strength of those interaction and those. So for example, it is that the provide level free among the level free measurements provide real time reaction kinetics to determine the dynamic parameters of bimolecular interactions and understands the protein and the function. Okay, so this level free measurements help us to understand in the real time manner. So if I say it based on the based on the inherent property of the molecule. Everything is based on the inherent property of the molecule, right? And it allows the measurement in. So it's nothing on the tag or something binding. It allows on the inherent property of the molecule. And the second thing is that it allows the measurement in real time manner. Okay, so it allows the measurement in the real time manner. So now, among which one type of technology is the carbon nanotubes or nanowires. So this is the kind of can. So these are some of like this. You can say that the nanotubes or nanowires. So it is like the hollow cylindrical graphite sheets and exhibits high level of chemical stability and the uh, mechanical strength and well-defined electrical and mechanical properties. So it has some inherent electrical and mechanical properties. Right, electrical and the mechanical properties. So now this is the application for the nanoscale biosensors. So now if you have those proteins or antibodies imprinted on it, and if or ligand whatever you have, if you have this protein binds to it, the changes in conductance. So this is change in conductance is measured. The change, this kind of changes in the conductance is measured so that if the protein has bind to the receptor or not is understood, right? Next is the ellipsometry, which is based on the polarization of the light. If you have this kind of beam and this light comes and the changes, so altered changes in the dielectric property or the refracting index of the sample surface. And so for ellipsometry imaging, you require ellipsometer, microscopy, then you have the CCD camera, etc., etc. So this is this measures the protein content on the solid surface. So one is using, for example, basic concepts of physics. Right, we are getting the basic concept of physics, correct? So one like the carbon nanotubes and nanowires. if we have the carbon nanotubes and nanowires as we have mentioned. One is using the principle of changes in conductance. When you have if the protein gets bind to its molecule, so you will have the changes in conductance. But in the ellipsometry, you are changing in the dielectric property or the refractive index. Changes in dielectric property or refractive index, right? So changes in the dielectric property, right? Changes in the dielectric property or the refractive index, that is in the ellipsometry. And in the carbon nanotubes or nanowires, which are the graphite sheets, you can measure the conductance change by the measure. Now, which is one of the major level-free techniques is SPR, or we talk about surface plasmon resonance, right? So what is plasmons? So plasmons are something that the special electromagnetic waves. Now again in the SPR or the plasmons, what we are trying to get is that the special electromagnetic waves that can be excited at certain metal interfaces, right? Like the gold and silver. So this creates are the, so plasmons are name so of SPR's surface plasmon resonance. Plasmons are something of the special EM waves excited at certain metal interfaces. Excited at certain metal interfaces of gold and silver, like gold and silver. So this is generated on the boundary of the metal and air, and it is very sensitive to change of this boundary of this metal and air, whatever this uh, uh, material whichever boundary is there. So this, in this boundary, the it is very much sensitive to any kind of change on those particular boundaries, and it leads to the adsorption of biomolecules to the metal. So now the problem in this is that not the problem, the major concept it is that the gold chip or the silver which can generate this plasmon. So if you have these particular ligands and if you have an analyte, so this with this boundary it is very much sensitive, whatever gets absorbed and it creates changes. So this gives us the concept of the sensogram or SPR sensogram, what it is known as. So what this particular thing is, you can uh, tell is that the changes in the SPR signal versus the time. So what it is there, that the light source getting the detector and it is getting the changed, right? So now if you have this baseline, so in the it occurs in real time, right? So you are getting the baseline when the molecule starts binding to it, association, then when you are uh, keeping the flow and it is getting out. So then after equilibrium, it will be the dissociation, then regeneration, and then finally uh, it is. So this is how you are getting the change in response unit, changes in response over time. So in the x-axis is time, and it is the response unit that you are measuring when it is getting absorbed on the surface of this cold chip, which are known as the plasmons, or the special kind of electromagnetic waves that can be generated over certain chips like gold and silver. So now different kinds of affinities and those things that can be determined, like kinetics of the rates of the reaction, affinity for the strength of binding, KD, or which is KD by K, but is the on rate and the off rate, which you can know the dissociation constant. So once you calculate the on rate, that is the association rate, and the off rate, you can calculate the dissociation constant, stating that what is the strength of binding of between these two ligands, or these two protein and the ligand. So this is quite important parameter to understand that, to study when we are studying the interactions. So what are the advantages of SPR? If we say in apart to the label based techniques or other techniques that we look upon, it is a label free, no need for tedious or expensive labeling controls. Again, you have seen that RPPA, we have to keep another slight control where we are uh, expensive labeled secondary antibodies, we are uh, flowing over them to get the non-significant, extract the non-significant signals informations out, or further normalizing those things, that okay, so which backgrounds to be removed, then to avoid the potential labeling artifacts. So that is also quite important. Then when we are saying that we have the direct, it is the measures the binding of the actual analyte. Okay, then it is a real time manner it happens. So it is direct and it helps to measure the binding of the actual analyte and to avoid potential any kind of labeling artifacts. So in real time, what it happens is that it allows the user to monitor the experiment, and obviously it is not an endpoint assay, and it measures the binding kinetics and the affinity. And then the limitations are that again you need to have a kind of homogeneous samples, difficult with the heterogeneous samples, difficult with heterogeneous samples, right? So it is difficult with the heterogeneous samples. Then sample preparation and the probe attachment to the metal surface might be difficult. So the sample preparation and to the metal surface, those attachments might be little bit difficult and can be tricky to some extent. Then the non-specific interactions of which we have no control over because we are measuring the real-time manner and whatever uh, those plasmons are getting and whatever response says we get, and over the time that needs to be, we need to have that. We need to ensure some specific signal and the bulk effects. So this is how this is happening, which you can also have given you one further reference for you to get uh, much deeper insights on the principle of the surface plasmon resonance and how it is actually working upon. So you can see that you have the sensor chip, which is the gold film or the gold chip, and and we have also described what is the meaning of the word plasmon, which is arise. So why it's only gold you can understand, because these plasmons are generated on the some metals like gold and silver. So we have the gold, we have the medium, we have the light, we have the gold coating, functional coating, bioregulation molecules and nanolites. And you can see that whatever adsorbed is happening, so we are getting the responses and we monitor the response units over time to get the sensogram, and we can calculate the KD and on rate and off rate to calculate the dissociation constant to understand how strong interaction between the two ligands are, or to the protein and the ligands, right? So now these are some of the examples case study which you can definitely try it out, like the KD calculation. The dissociation constant is a measure of the strength of the interaction between your protein and the ligand from the association and the dissociation rate constants. So KD equals to KD by Ka. So if K equals to this much and KD is, then KD equals to 4 nanomolar, right? Second is the binding kinetics. So the association and the dissociation resistance can be a sphere in different models. So this is for the fitting of those kind of models. So for example, K equals to this much and KD. So the KD of 16.7 nanometers. So you can try it out and with their examples on your own for a better read. Then for generate concentration response curves for ligand binding to a protein, which can be KD. Concentration of small molecules show the responses linearly with concentration train, after which it plateaus, indicating saturation of the binding sites, KD of 500 nanometer. Then competitive binding assays on binding of primary ligand. So this all you can practice. Now the second examples are like you want to immobilize a small molecule ligand on an SPR sensor chip and use it to study to binding to a protein. Rmax value of 2000 RU, that is the response resonance units, and have some preliminary data like the KD is this much 50 density. So this is the formula how we will be doing that to response Rmax and to molecular weight into Na by, and then you are giving the NO is Avogadro's number, is the thickness of the adsorbed layers, and finally you can uh, get the value of the ligands that you need to mobilize in that kind of sensor chips. So these are some of the examples. So in the, so whatever we have started with the level based technologies like the reverse phase protein arrays, and then of last portion of the level based techniques like the reverse phase protein arrays, and before that we had done NAPA, and here we had seen the wide variety of the samples to be used, but different kinds of antibody checks, validation, and that is a tedious process as well as a costly process if that antibodies does not work out, and which antibodies can be taken forward with different four control checks, and then finally you are getting the result and the labels and finally the data analysis technique. So now moving on is label free techniques, which is does not require the TDS and those non-specific binding, and can be further taken on in an easy manner with the uh, concept of what we can talk about, or what we talked about is that in the real-time manner we can monitor, among which there are carbon nanotubes or carbon nanowires, like the carbon nanotubes and nanowires, which are the graphite sheet and changing the conductance, or ellipsometry, which is changing the ellipse layer, that is a refractive index or the dielectric constant, or the surface plasmon resonance, of which the plasmons are generated over a particular metal like gold or silver, and you are measuring the real time monitoring of the measurements of the as you are getting the responsiveness. So, and some case studies have provided so that you go through it, and this is all. Yeah. So, if there are further no doubts, so yeah, thank you, and then we can meet about the uh, next